Good evening to all the CUET aspirants. So here we are with the concept of cell membrane. We have learnt is about completely prokaryotic cell, and we have done with the cell wall. Prokaryotic cell introduction, difference between plant cell and animal cell, followed by the limiting boundary of a plant cell is completely done, that cell wall. How cell wall formation is done, what is the mechanism of cell wall formation, composition of cell wall, everything is dealt. Today we are going to deal with the limiting boundary of an animal cell. What is the chemical composition of cell membrane, followed by different models of cell membrane. Then we will also look into the protein structures and functions of the cell membrane in detail, followed by first we look into the double membrane bound organelles, mitochondria and plastid, mitochondrial DNA, plastid DNA, followed by the mechanism of replication and pattern of inheritance involved. And then post which we will also deal with nucleus, nuclear DNA, replication methods, and then about plant. So in today's concepts, what we are dealing is about cell membrane, its models, functions, and then okay. Let's focus here. The chemical composition of a plasma membrane, of course, it includes lipids, protein, and carbohydrates. Okay. If I have to consider RBC cell membrane as a concept, RBC cell membrane, whose RBC cell membrane, if I have to consider mosaic model given by Singer and Nicholson, 52% of proteins, around 50% of proteins, around 40% of lipids, and around 5 to 8% of carbohydrates will be there. And this carbohydrate composition is also in the form of glycoproteins and glycolipids. Okay, it is in the form of glycoproteins and glycolipids. If you look into the thickness, the general thickness of cell membrane is around 75 angstroms to 150 angstroms. Okay, and it varies in different cells. Based on cell, the thickness of cell membrane will be varying. Electron microscopic structure, that is simply said, cell membrane is having a trilamellar structure with outermost protein layer, innermost protein layer, where protein layer thickness is around 20 angstroms each. Lipid layer is in between, which is sandwiched in between the protein layers, and the lipid layer thickness will be approximately 35 angstroms. Clear? Fine? Any doubts? Now is the voice clear, ma? Yeah. Next, quantitative analysis of plasma membrane. Look into the quantitative analysis. Proteins and lipids are present in the great amount than carbohydrates. Okay. Chemical composition of plasma membrane includes mainly what? Proteins and lipids. As we have already discussed, approximately 40% of the cell membrane composition is made up of proteins, of lipids. It is made up of lipids. Around 50% present. So here if you see, what are the types of lipids that are present over the cell membrane? Types of lipids include majorly phospholipids, glycolipids, sphingolipids, and sterols, which are cholesterols. Okay. Sterols, which are cholesterols.
About 55 mm thickness.
The major composition of these four types are phospholipids. Phospholipids will be having a polar head and a non-polar head. Polar head is known as hydrophilic polar head. Hydrophilic polar head, and this is hydrophobic non-polar tail. This hydrophobic non-polar tail inside the plasma membrane is protected from this aqueous environment of the extracellular space by what? Hydrophilic polar head. Once again I repeat, the hydrophobic non-polar tail, which is towards the plasma membrane, is protected from the outer aqueous environment by what? Hydrophilic polar head. Okay. So each phospholipid is having a head portion and a tail portion with hydrophilic phosphate molecule, which is having negative charge. Remember, the hydrophilic phosphate molecule has what? Negative charge, and inside it is having hydrophobic fatty acid moiety. In inner side it is hydrophobic, outer side it is hydrophilic polar head. Clear, right? Such molecules are known as amphipathic molecules because it has both hydrophilic head and hydrophobic tail. Fine, right? And then phospholipids are arranged as bimolecular layer. Okay, it is bimolecular layer in the membrane. Acidic lipid parts binds with negatively charged protein parts. Okay, fatty acid chains are present, right? That acidic lipid parts will bind with what? Protein regions which are negatively charged. Fine, right? No? And then, yeah, here you see, now you have understood the composition of lipids. What type of lipids are present? Phospholipids, glycolipids, sphingolipids and sterols. Sterols are cholesterols. Cholesterols are to maintain the fluidity of the cell membrane. Amongst all these four types, which is widely present, which is
volume means
phospholipid is dominantly present. Now we will look into different types of plasma membrane proteins which are present. If you can remember this one table, you can answer any question that is coming from membrane proteins. If you want, you can take a screenshot also. Fine, and then remember. So you have different types of proteins now. Structural proteins, functional proteins, carrier proteins. Structural proteins based on the ease of extraction are again of two types. Okay, those are peripheral or extrinsic proteins, integral proteins and transmembrane proteins. Okay, structural, functional, carrier. These are the main categories of proteins based on their functions which are on the plasma membrane. Structural proteins are again of three types based on the ease of extraction. Peripheral proteins can be easily extracted by the use of some detergents, whereas integral proteins which are deeply embedded inside the lipid bilayer, they need the action of some strong acids for the extraction. Transmembrane proteins will transpan either one side of the lipid bilayer or both the sides of lipid bilayer. That is the reason we say as integral proteins will definitely transpan two sides of the lipid bilayer. So we say all integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Remember that point, important for statement based. So here if you see peripheral located on the outer or inner surface, this is embedded in the lipid bilayer, transpans any of the sides or both the sides, transmembrane protein. This is loosely attached, firmly attached, this is also firmly attached. What is the main role of the structural extrinsic protein? It's a support, it maintains the membrane shape and it interacts with cytoskeleton structures. What are the cytoskeleton structures? Microtubules, 18 filaments, intermediary filaments. These are different cytoskeleton structures. So, the peripheral extrinsic protein interacting with the cytoskeleton structures, it will maintain the integrity of the cell. Okay, and here you see the examples: surface enzymes, receptor associated proteins. Clear, right? And now about integral proteins. This is firmly attached with what? Hydrophobic core. Okay, mechanical stability and it will maintain the structural integrity. What is an example? Membrane anchoring proteins are the examples for intrinsic. Next, transmembrane proteins. These act as channels, receptors over the cell membrane, and they are also useful as mechanical support. Okay, the channel proteins and receptor proteins are the examples of transmembrane proteins. Clear with structural proteins, both extrinsic, intrinsic and transmembrane. Now we will move on to functional proteins. Will mostly play enzymatic role, that is catalytic role, which is happening inside any metabolic activity of a cell. So enzymatic proteins, if you see, they are usually surface or they are embedded deep inside. Nature of association, they are associated with lipids or protein components. Main functions include they catalyze biochemical reactions which are happening at the membrane. Example, enzymes like acid phosphatase, alkaline phosphatase and RNase. These are the enzymes which are associated. Clear, right? And then you see carrier proteins. These are anaerobic proteins. Example, permease. Okay. Location, they are embedded deep inside the membrane. Specific binding sites of the solutes they are, and main functions include they transport ions, molecules all across the membrane. Examples include galactoside permease. Okay. And then carrier proteins include unipods, sympods and antipods. Their location is all across the membrane. Fine, right? Transports specific configuration of the proteins only.
Now this is in seconds.
Specific configuration which will fit into that carrier proteins. Active and passive transport, both the modes of transport it will facilitate, and these are what examples include ion transport proteins. Clear with this table? Any doubts anywhere?
Two, 1, 3, 6. Okay. Yeah. So now just look any of.
Okay. And now. So we said carbon is so present over the cell membrane. First you have to know how carbohydrates are associated, either in the form of glycoproteins or in the form of glycolipids. If glucose units are associated with glycoproteins, sugar molecules like oligosaccharides, they are attached to what? Either peripheral proteins or integral proteins. If polysaccharide units are associated along with lipids directly, attached to the heads, hydrophilic polar heads of the lipids. Hydrophilic polar heads molecules. If they are associated like this, glycoproteins and glycolipids. What is their main function or the main role? Main function is cell to cell interaction or attachment. Cell to cell interaction is the main function that is performed by the glucose units which are present over the cell membrane. Understood ma? What is the main function? Cell to cell interaction or cell to cell association. Now you look into the ultrastructure of cell membrane. Many models are being proposed to explain the plasma membrane model after knowing out the composition of the cell membrane. So here you see important models include Danely-Davison's model, Sure. I hope now the voice is clear. Yeah. And then we continue with the models that were proposed to explain the plasma membrane. After knowing the composition of plasma membrane, proposed what? Danely-Davison's bimolecular model, which is also known as sandwich model, and then Robertson's trilamellar model, which is also known as unit membrane model, and then Heeler and Hoffman's micellar model. Okay, after which accepted model, which is proposed in around 1972, Singer and Nicholson's fluid mosaic model. Okay, and now I'll anyhow send you the PDF. Okay. Yeah. Here we go with Danely and Davison's model. Make sure you are understanding everything. If having any doubt, you ask me. Okay. For content and all, I'll send you the PDF. Yeah. This is proposed bimolecular model of plasma membrane structure. It has two layers. Okay. Lipid layer is sandwiched in between the globular protein layers. Can you see here how lipid layer is sandwiched? See, lipid solution center is sandwiched by globular proteins on the other ends. This is bimolecular model because two molecules are included, that is lipids and proteins. So in the middle form, so layers cover the lipid layer on the both the sides. Sandwich model. This is also known as sandwich model because lipids are being sandwiched by proteins. Yeah. So here you see exterior side protein layer, middle you see the lipid bilayer is present, interior side again you will have protein side. Okay, presence of lipid lined protein lined pores are present on both the ends. You can see protein lined pores are present. These pores, what are transported to the either sides? Water ions are transported
in
either sides. Clear, right? Any doubts anywhere, ma?
Use this.
And then Robertson's trilamellar model or unit membrane model. Based on the earlier model, which is called as unit membrane model, because the central structure, that is lipid bilayer which is present, is again having what? Protein on the either ends. But here you can see lipid bilayer at the center. Okay. Previously, if you see in the sandwich model also, it is there as a unit on whole. Here it is clearly explained that 3-layer structure. That is explained based on the concept of sandwich model, that lipids are sandwiched by 2 protein layers, but this is explained on the concept of complete unit membrane. Completely the membrane is explained as what? A single layer. Okay, as trilamellar model, 3 layers are present. Here this is what the unit membrane. Central lipid layer is of 35 angstroms thickness. On the either it has what? 20 angstroms of thickness. Nature of interaction, if you see, electrostatic forces will exist between them. Okay, positive are interacting with negatively charged components.
Okay, R is
Hydrophobic fatty acids.
okay. So 1 by R is what? G. Okay.
The center maintains what?
1 by 60 you can
And here mucoproteins are present on the outer margin. Non-mucoproteins are present in the inner margin. Major point to be addressed. What type of proteins are present on the outer and inner margin? On outer margin it is mucoproteins. On inner margin it is non-mucoproteins. Robertson also called it model as what? Greater model. Trilamellar, unit membrane, greater model. What is the outer protein composition? What is inner protein composition is important, right ma? Next. Hiller-Hoffman's micellar model. He explained the model in the form of micelles. Okay, the outer protein layer is covered, inner it has what? Lipid layer. These lipid molecules are arranged in the form of micelles like this. So from the diagram itself I will explain you see. Here can you see the micelle? Outer is covering the entire micelle. If you see the center,
if resistance is not mentioned,
acid chains are present. At the periphery ends of the mycelium, what are present? Hydrophilic lipid molecules are present. The mycelium on total or on whole, it is covered by what? Protein structure. At the lipid is there. Lipid also, how is it arranged? Lipid molecule, phospholipid here arranged like fatty acids, hydrophobic fatty acid molecules at the center, at the periphery. Like that it is forming a mycelium. And this mycelium on whole it is surrounded by what? Protein structure. So here you see the
these are 4
lamellar model according to this.
What this?
Yes, it doesn't have a lamellar model. Membrane consists of globular
between K place
and the
N place. It is having more conductivity of
40 to 70 angstroms. Each mycelium will contain how many phospholipids? 6 to 8 phospholipids. Single mycelium. Okay. Hydrophobic
K. So conductivity value for K is more now. So that's why this line is above. That's it. Next one.
Water-filled layer pore region. This water-filled pores are called as what? Model do not widely accepted.
And one more is CuSO4.
Structures. That is the major drawback. Theoretically it is, hypothetically it is accepted, but in reality it doesn't work. It is not the model by which is acceptable for the living membranes. Okay, this is about Hiller and Hoffman's micellar model. Now we will go through the most widely accepted model, that is fluid mosaic model, which is proposed by S.J. Singer and G.L. Nicholson. This explains the structure of plasma membrane and biological membrane. It's widely accepted model. The reason for acceptance, if you see this structures what? Electron microscopic.
1 farad. That's why.
The main reason this is widely accepted.
So only 1 farad is required. So what is the mole ratio?
To the practical zone. This explains membrane functions based on the arrangement of components. Plasma membrane has fluid consistency, that is because of lipid composition, and protein is distributed all around in the mosaic pattern. So here first we will learn about the model and then we will go with the theoretical explanation.
Normality, what's the formula?
Here we go with the first. Yeah. Lipid, can you see? Same model wherein hydrophilic head, polar head is towards the outer side, hydrophobic non-polar tail is clear, right? The proteins are of 3 types. Peripheral proteins which are on the outer side, integral proteins which are
grams we are saying number of
both the
sides given
plasma membrane. You can see the transmembrane proteins which are transparent of the sides of the plasma membrane. In that case, all integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Clear, right, about 3 types of proteins. Now you see about glycoproteins and glycoproteins.
Here, decimolar, no? 1 by
There are some
so weight will be no?
joining proteins also there are some oligosaccharides that are present, which are around 8 sugar molecules. These are called as glycolipids, and these are what? Glycolipids. What is their main function? Cell to cell interaction. So just imagine like how proteins are present over the lipids. Lipid molecule is like quasi-fluid structure, like this quasi-fluid structure. Okay, over the lipids, these proteins are like icebergs on the sea of lipids. Yes or no? This protein seems to be icebergs on the sea of lipids, and this quasi-fluid nature of the lipids will allow the lateral movement of proteins. Okay, over the bilayer of the lipids, whichever proteins are present, the proteins will be moved laterally. That is because of what? Quasi-fluid nature of the lipids. This will also allow the flip-flop movement of proteins as well. Okay, from one lipid layer to the other, and also allow the lateral movement. Alright, yeah. Arrangement of lipids. Lipid molecules are arranged in 2 rows. Hydrophilic heads, hydrophobic tails. Clear to us about lipids, right? Now, look into the types of lipid movement.
This as a function of nature of gas.
That is quasi-fluid nature. It is not static. Dynamic means it is always changing. Fluidic and it will always be changing. Lipids are not static. They are not stable. They are dynamic. They replace in the constant
Gas.
Okay. Types of movements you can see flip-flop movement. Lipids move from one layer to the other layer. They will move on the lateral diffusion. Lipid exchanges position with the neighboring rotation. Tail part rotates axis. These are the movements of what? Lipids, which make the plasma membrane to be dynamic in nature. Now you see functions of the plasma membrane. Okay. About proteins we have already learnt in the functions of different proteins table. Lipids we have learnt here. Now we look into the functions of plasma membrane. Mainly of what composition? Lipoproteinaceous composition. Yes or no? This performs various plant cell. General functions: shape and stability to the plant cell, regulates flow of water, solvents, mineral ions, gases to the cell, and this regulates outward movement.
It is boiled at 100 degrees. Now it is elevated for 101, 102 like that.
First you see transfer molecules and substances. Plasma membrane is also noted as semi-permeable membrane. Okay, it is addressed as semi-permeable membrane because it will allow the movement of solvent molecules and it will select which solute particles to allow and which do not. That is the reason it is addressed as what? Semi-permeable membrane. And this allows only water molecules to pass, not solute particles. The entry of water molecules inside the cell is known as what? Endosmosis. Okay.
With a greater value, what do we have?
Plants
3 ion,
semi-permeable nature, and few gases are also permeable.
0.6.
Like what? Carbon dioxide, oxygen. They diffuse all across the plasma membrane. That doesn't require any facilitated diffusion, any carrier proteins. It doesn't require anything as such. They are diffused like that. Okay. Clear endosmosis?
And this one?
About the term of semi-permeable nature.
Totally.
Yeah, and it is also known as permeable membrane. It is addressed as selectively permeable membrane because it selects which solute particles enter into the cell. Okay, so selectively permeable, semi-permeable, and as it allows water molecules, that phenomena is called as what? Endosmosis. Next, about ionic occurs by two absorption. One is what? Passive absorption, and second is active absorption. Passive and active absorption.
One is highest.
Passive and active absorption. Here you see, is it clear now?
Just duplication and if I ask for
Is the voice clear now? Yeah. Please let me know about the voice clarity, ma. I'll continue with the class. Yeah.
deviation.
Accumulate what type of ion? Sodium and potassium ion.
No,
Against the gradient. Fine, right? When it is against the gradient, it
it
requires
should be 80.
carrier proteins. Like if the carrier protein transports only single solute, then it is known as what? Uniport. Two molecules in the same direction addressed as what? Symport. Molecules in the opposite direction known as what? Antiport. Okay. So you have uniport, symport and antiport. ATP transporters, which is also known as ATP binding cassette. Transport materials, this uses what? Energy to transport the materials, especially toxin substances to move it outside the cell continuously. What is ATP binding cassette? By utilizing the energy, the molecules are being continuously excreted out of the cell. They are moved out of the cell by what method? Exocytosis. Clear till here, ma, about carrier proteins? Yes, any doubts anywhere? Yeah, and then now here you see about endocytosis. Here cells will import or export large molecules across the cell membrane. How they are importing large molecules? Via two methods. One is known as pinocytosis or receptor mediated endocytosis. Okay. Pinocytosis is also known as cell drinking. Pinocytosis is also known as eating. Okay. Types of noted. Two types of endocytosis. One is known as which is known as cell drinking, okay. So liquid droplets are not transported through the membrane, are taken into the cell. They are taken inside the cell. Plasma membrane surrounds the substance completely. Okay. Then the material is swallowed into the cell. What are the molecules that are being entering into the cell via pinocytosis? Amino acids and some proteins. They are entered into the cell as liquid droplets. Okay. Cell will be engulfing those liquid droplets. That is known as what? Pinocytosis. Clear, right? For example, just a second. These are the lipid droplets we have. If this you observe like a cell, this engulfs the lipid molecules like this. Okay. This is known as what? Pinocytosis. Same ways here you see. Receptor mediated endocytosis. The procedure is entirely same, but here receptor over the plasma membrane is involved to accept the molecules as ligands. Whichever molecule that was entering the cell, that molecule acts as ligand. And a specific receptor, this ligand will fit into a specific receptor to the cell. This is known as receptor mediated endocytosis. Look at here carefully. Specific receptors on the plasma membrane will recognize macromolecules which are present outside. The attachment of the substance is called as what? Ligand. That will bind to what? Receptors. Those ligands can be anything. It can be a virus, it can be protein, it can be anything. The ligands will bind to the cell surface receptors. This is ligand loaded receptors. They will move into the membrane pits. They will move into the membrane pits like this. Okay, and the membrane invaginates inwards and then it binds up. Vesicle will patch off into the cell lumen. See, this is completely cell lumen. A receptor is present that is accepting the ligand. A small vesicle is formed once the ligand is being accepted. This forms a vesicle and this pinches off. Okay, and this vesicle is completely transferred into the cell lumen. Okay, fine, right? Receptor mediated endocytosis.
Yes,
yeah. Next is what is exocytosis. Exocytosis is also known as cell vomiting. Endocytosis of two types. Pinocytosis, endocytosis is cell eating, and exocytosis is cell vomiting. Okay, so remember the excretion of macromolecules, especially some toxins or dead particles which are present inside the cell, are omitted out via effaggy or exocytosis. This is also known as effaggy. Okay, and about phagocytosis I didn't tell you, right? Second type of endocytosis. The mechanism is completely same, but whenever solid particles are entering the cell, they will enter as a phagosome, which is a globule. Okay, they will enter as a phagosome attached to the receptors of cell surface. Okay, via cell receptors, they will enter inside the cell as phagosome. This will be lysed by the lysosomes, which is known as phagolysosome. After utilizing the molecules, it will be excreted out. Mainly this is for solid particles, like whenever some food particle enters inside the cell. To digest the food particles, lysosomes will be rich in the hydrolytic enzymes. Tell me some hydrolytic enzymes that are present inside the lysosomes. Any hydrolytic enzymes?
So NB is what? X modes of B.
Hydrolases, these are enzymes. They will attach to this phagosome. Let's say it is having some food particles inside it. Okay, attaching to this lysosome will completely digest phagosome. Fine, right? Which is known as phagolysosomes, or secondary lysosomes also we'll call it as. After digestion, the molecules whichever are not required by the they are omitted outside as what? Soluble debris by the phenomena of exocytosis. Clear till here? Any doubts? Any? Yeah, and then
Hydrogen comes into.
now we will start out with double membrane bound organelles. We will start with mitochondria. Okay, mitochondria, which means what? It is popularly known as powerhouse of the cell because it is synthesizing ATP molecules, which are the energy currencies of the cells. But the term is first given by or used by Benda in the year 1897. Mitos means thread, chondro means granule. If you look into the components of mitochondria, mitochondria will have an outer mitochondrial membrane, which is outer mitochondrial membrane, and inner mitochondrial membrane is folded into cristae. Inner mitochondrial membrane is folded into cristae. Outer mitochondrial is soluble. It is permeable. Semi-permeable. It is selectively permeable. Okay. And this inner mitochondrial membrane will differentiate the matrix into two parts. Okay. Whatever the region that is present inside the cristae, okay, is called as mitochondrial matrix, and the space in between cristae and outer mitochondrial membrane is known as what? Intermembrane space. Clear with the components of mitochondria? Okay. For complete metabolic activity to happen inside the mitochondria, whatever enzymes, protein molecules, carrier proteins, okay, whichever are required, they are present in the inner mitochondrial membrane only. Whatever protein molecules, channels, carrier proteins are required for the metabolic function of the mitochondria are present where? They are located inside the inner mitochondrial membrane. And matrix of mitochondria will have double-stranded circular DNA, and it also has 70S ribosomes. These two features will show a strong resemblance with prokaryotic cell, okay, possessing double 70S ribosomes will show a strong resemblance with what? Prokaryotic cell, which is clearly explained by endosymbiotic theory. Both mitochondria and chloroplast on a time, both of them were individual cells, but in the due course of evolution, when eukaryotes started evolving, they engulfed the prokaryotic cells. That is, mitochondria and chloroplast are being engulfed by the eukaryotic cells in the due course of evolution, and later what happened is they became part and parcel of the eukaryotic cell, okay, but still they emit the prokaryotic cell features like what? Double-stranded circular DNA having 70S ribosomes replicating by binary fission. These are the features that it possesses, okay. So the general features of mitochondria which it has is cylindrical or rod-shaped bodies. These are whose diameter is around 0.2 micrometers and the length it has is around 3 to 10 micrometers. The shape of mitochondria is completely dynamic, which will fuse and then they will divide. Fine, right? They arise by what? Fusion of pre-existing mitochondria, and they divide by binary fission. Fine, right? Number per cell. How many mitochondria are present per cell? It can be around 200 to 300 mitochondria per cell, or around 500,000 molecules in some protozoans like cows. This protozoa in a single cell, how many are present? Lakhs of mitochondria, 5 lakhs of mitochondria. This is basically of mitochondria organelle. Clear, right, ma? So now whatever we have learnt about the structural description, look at here. Outer membrane thickness is around 60 angstroms. It is smooth and regular. It contains some porine proteins, okay, which allows the passage of molecules around 5000 Daltons. Lipids, fine, right? It is completely permeable and it is having porine proteins. And it has some irregularly shaped lipids called as cardiolipids, okay. And here in the inner membrane, whose thickness is 60 angstroms, located how much? 20 to 60 angstroms away from the outer membrane. Okay, okay, this is highly selective and impermeable to ions. This is folded inwards like what? Cristae. Cristae thickness will be around what? Inner mitochondrial membrane thickness is around 140 to 180 angstroms. This houses electron transport chain, which is ATP synthase molecule, which is required for the synthesis of ATP.
When it is possible,
B
by reverse osmosis.
Towards the mitochondrial matrix. Aid in the synthesis of ATP. They are also known as ATP synthase or oxysomes or Fernandez-Morgan particles. Fine, right? Or if not F1 particles also we'll call it as. Okay. Next is intermembrane space. This is a space between outer and inner mitochondrial membrane where complete action of electron transport system takes place. Of course, it is operated in inner mitochondrial membrane, but then proton gradient is created here in the intermembrane space. Next is about matrix, which is the inner chamber. What does it contain? In the contains what? Krebs cycle enzymes, fatty acid oxidation enzymes, pyruvate oxidation enzymes, RNA and glycolysis and cytoplasm. Very different. Glycolysis is the common respiratory pathway for aerobic and anaerobic respiration, which is operated in the cytoplasm. Okay, from cytoplasm, see, as you asked, I will explain here. See, for example, consider this as one cell.
Yes, ma'am.
This as one cell. Let us suppose a eukaryotic cell. Fine, right, ma? Here complete cytoplasm is present. Let us draw a rough diagram of mitochondria. Fine, right, ma? Let us suppose this as outer mitochondrial membrane. Okay, this as inner mitochondrial membrane, and this will be what? This will be what? Intermembrane space, and this will what? Mitochondrial matrix. So here glycolysis will take place here in the cytoplasm. This is cytoplasm. Glycolysis occurs here. Fine, right? Wherein the glucose molecule will be converted into two molecules of pyruvate. Okay, and the net ATP released into the cytoplasm will be 2 ATP, and it will also release 2 NADH. Fine, right, ma? This pyruvate will directly enter into the mitochondrial matrix. From cytoplasm, it will directly enter into the mitochondrial matrix. In mitochondrial matrix, what happens is, first process that happens, oxidative decarboxylation, and second that happens is Krebs cycle. Okay, at the end of oxidative decarboxylation and Krebs cycle together, okay, for one glucose molecule, how much amount of energy is released? It is, for one glucose molecule I am saying, it is 8 NADH, 2 FADH, 2 FADH2 and 2 ATP. Fine, right? And the ATP whichever is released out here will be utilized, but the NADH, to get oxidized again, it has to enter matrix, right? How it will enter? It will enter via shuttle systems, which is known as glyceraldehyde 3-phosphate shuttle system. For that, for one NADH to transport, it will utilize 1 ATP. Okay, the transport charges are 1 ATP by this glyceraldehyde 3-phosphate. Like that, it will be transported into the matrix. Now, inside the matrix, it is powerhouse of what? NADH, FADH2. Now, this complex energy molecules should be simplified to what? ATP, because ATP is the energy currency of the cell. For all the metabolic activities inside the cell, the energy is utilized only in the form of ATP. So, it has to be simplified, the energy has to be simplified to ATP. Fine, right, now? So, that process is done via electron transport system, which is last process of aerobic respiration, which operates inside the inner mitochondrial membrane or cristae. That we will discuss tomorrow. Okay, from mitochondria, what parts are left is electron transport system, we will deal it tomorrow, and the second is mitochondrial DNA and its maternal inheritance, and the diseases which are caused if the pathways are inhibited from mitochondria, post which plastid cell, plastid DNA and nucleus also we will do it tomorrow. Okay, so I hope today's class is clear, right?
Yes.
Yeah.